What is DNA? Well, for starters, DNA is a complicated double helix that stores all our genetic information. Think of it, in a sense, as a blueprint for the body. DNA is made up of phosphates, sugars, and bases, all held together by hydrogen bonds. There are 4 different types of bases in a DNA strand: thymine, cytosine, adenine, and guanine. Thymine will only be found next to adenine, as cytosine will always be found next to guanine. Sugars contained inside of DNA contain no oxygen, making it contain deoxyribose sugars instead of ribose sugars. mRNA is a little different than DNA. Though both are located in the nucleus, mRNA moves into the cytoplasm in a process called translation, but we'll get into that later. mRNA is like half of a DNA molecule that works to transcribe and transmit DNA to the ribosomes to create proteins. While DNA contains thymine, cytosine, adenine, and guanine, RNA contains uracil instead of thymine. Transcription is when DNA is copied by RNA inside of the nucleus before the mRNA leaves the nucleus to perform translation. After transcription, translation occurs. Translation is when mRNA connects to the ribosome to create proteins. After this connection, tRNA brings in amino acids to build the proteins. Codons on the mRNA strand connect to anticodons on the tRNA strand to show the order in which the amino acids need to connect to create polypeptides. Amino acids can combine with peptide bonds to create a polypeptide bond. Polypeptide bonds are then folded in one of 4 ways to create a protein: primary, secondary, tertiary, and quaternary. It operates, becomes RNA, as well as uses its blueprint of genetic makeup to create proteins, which in turn will create a living being.